more question, and maybe we can take some questions from the audience, but we had talked about, and earlier today as well, is how do standards need to be set here? There's multiple platforms available to do sequencing. Where do we need to be concerned about quality, and how should those standards be set? George?
Well, so to some extent there are standards that, and they're very important in that if the FDA or the General Accounting Office does a test where they send an identical sample to a bunch of different companies and they get 5 different answers for 5 different companies, they're not going to say, oh, company number 1 is right and all the others are wrong. They're going to say they're all wrong, basically, because there's no consensus. So you need to have, even if you don't share every genome, you need to share some basic level of genome interpretation. And we have some of that around. There is an app for that. There are a few of them. We make our algorithms publicly available, not because it's the best, but because it is critiquable and it is shareable. And then if anybody can add value onto that, but at least that can be the ground truth which the FDA can test us all on. So I think that's extremely important. And so we have a standard of sorts there.
And is that being used broadly? The standard that you've set, is that broadly used?
Well, I mean, we're still at early stages. Right. Not everybody in the room got their genome sequenced, so it's not broadly used.
Tomorrow.
But if they did, yes, it would be broadly used.
But you can get a CAP-CLIA certification, which is clinical licensing for being able to do these tests. You absolutely can do it. We've done it. It's hard work. But the whole point of that is it reproducible? Can you make this reproducible? And the answer is yes. Now, can you always improve it? Will it get better with time? Absolutely, and there's issues around that. But absolutely, we need standards. We have to follow it. But they're in place. The CAP-CLIA guidelines is where we go. And so if you want to practice medicine with this is how you do it.
My concern is less the standards on the practice of medicine, because there's a great track record there, but more changing the rules for the discovery phase, because we still have a huge gap in our knowledge. Again, you've been focusing on some cancer cases or some N-of-1 pediatric cases, but we really do want to understand autism, schizophrenia, hypertension, these other cardiovascular diseases, and we do need the researchers around the globe to have some standards of sharing and maybe impose on them some requirements for sharing, because there is going to be a period of time over the next decade where we really do have to do big studies to create a lot more value to this information. And that's what I'm most concerned on. So if I see people who say, I had my genome and I only learned about a few dozen things, why can't I learn more? And I said, well, you can. After we do that next million, we aggregate that data and we do that research. So I do think there could be a tough time here, George, before everybody runs out and gets sequenced. There are a lot of people that I talk to who aren't these very special cases who kind of don't want to buy the Blu-ray until there's more content.
Right.
And to get that content, I do see huge problems right now. I watch all these people with underpowered studies, to use a technical term. Not enough data to get a good answer.
Well, perhaps in addition to our incentives to innovate, we'll add incentives to share and figure out a creative way to do that. Are there questions? Sure.
Chris Conover, Duke University and Forbes contributor. If this is so cost effective, I'm curious why a Kaiser health plan doesn't go ahead and sequence all of its patients or all of its newborns or something like that. And wouldn't that solve a lot of these problems? They had the patient data, they had the data on utilization and outcomes and all of that stuff. It seems like that sort of approach would quickly jumpstart this. Again, if it's truly cost effective and there's enough actionable material in these sequences for them to save money.
I can actually answer the Kaiser question because about 2 years ago, I was sitting down with the president of Kaiser, and he wanted to do a big SNP study, a genotyping study. It's kind of like thinking you understand a book by pulling out 100 words from it. And now we literally want to read the whole book. So I think the answer to your question is things have moved so quickly that if you put together that same group at Kaiser who said, let's genotype, let's look at 100 words in a 600-page book on 500,000 patients, I'm sure right now the conversation is, let's sequence those patients. And you will see groups like Kaiser doing it.
But I would say that you hit the nail on the head. I mean, the limitation here is people are afraid to jump in because what is the evidence of the economic value? There is no evidence, right? There's a handful of studies. And so everybody's waiting to see whether or not it's economical, but it's hard to imagine having your genome, which, by the way, is an accurate form of your family history. You know that form you fill out every time you go to every doctor, that you really don't know the diseases in your family? Why do we do that? Because it has value. But we're not ready economically. No one's shown this. So we're kind of waiting to see the chicken or the egg with data that is hard to imagine it not being useful.
If we need another stimulus program, I think this is a perfect opportunity.
I think it's going to happen without it, but it'd be nice to have it. You know.
Question?
Hi, Jeremy Grushko. I'm partner at Norton Rose in the Pharmaceuticals and Life Sciences Group. So maybe not the right question to follow up on that set of answers, but when we reach the point where there is enough data and the economic value of this is established, which I think we all think is coming, I'll ask in a concrete way, would you advise your kids to do pre-implementation genome sequencing before they have your grandkids?
I would recommend that they get preconception because your carrier status is very telling, and it's less less technological than say doing in vitro fertilization, and it's less jarring than doing abortion. So I think preconception is really the best time to do it, and everybody who is of reproductive age should be doing that today, not 10 years from now, today.
These are really personal questions. The reason I think it's called personalized medicine is because this is personal, right? So before I had my genome sequenced, I had a discussion with my children, right? What information do we want to know as a family? I mean, you really change the dynamics of this. But my daughters, there's breast cancer in my family, my daughters have a choice in terms of what information do they want. And they can always change their mind. And I think the key is to never make an assumption about what choice somebody should have to make. The goal is to enable people to make choices that's right for them. That's personal.
Okay. Other questions? Good. Well, I'll maybe close this out then with one for each of you to maybe think about for just a second. And I'll quote Albert Einstein in saying, if at first the idea is not absurd, then there's probably no hope for it. So my question for you is, what's your most absurd idea today? And what do you think the barrier for that is? Jonathan.
My first absurd one was high-throughput sequencing. Yeah. Wow. We're supposed to share the next one?
Yeah, what's the next one?
I don't think I'll be invited back, but I'm working on a tricorder. Remember Star Trek?
Yeah. Okay.
The day your child's born, you get your Apgar score and their genome.
That's inevitable.
And how far off
I think
is
that's
that?
not
That's
quite
today.
absurd enough, yeah.
Yeah.
But, I think I'm not working on tricorder, but I'm certainly working on portable sequencers. And more importantly, we're working on gene therapy. And it's already the case that there are people that are getting therapies where you take their stem cells out of their body, you engineer them, put them back, and they become, for example, resistant to the AIDS that's already in their body, so they become fully AIDS resistant. This is highly targeted gene therapy where you can change individual base pairs. I think we're going to see a million-fold decrease in costs for that, just like for the sequencing, and it's going to be incredibly accurate and powerful.
Right.
So George wants to write DNA as well as we read
it.
DNA.
Exactly. That's the next revolution, and it's not so far off.
Yeah.
Just like sequencing came down in price 50 years ahead of schedule, I think the same thing is going to happen for gene therapy. So genetics is no longer our destiny. We can change our environment, and we can change our genes.
Genes. Steve?
My most absurd idea is that it's no longer absurd for any well-informed patient to have a lot of leeway in deciding what kind of care they want to pursue along with their caregivers.
Great. Well, I don't know if all of you feel like I do, but I would classify it as maybe optimistically overwhelmed at this point. These have been great discussions today, so I thank the panel for coming and for your wonderful vision and thoughts and for the great work that you're doing on behalf of all of us and every patient that benefits from your work. Thank you.
Thank you.
Yeah.